Hi, I'm the one from Ms. Johnson's AP Biology class, and I will be discussing the cell cycle and regulations of the cell cycle. First of all, if the cell cycle is not regulated, cells could constantly undergo cell division. While this be while this may be beneficial to certain cells, the whole constant reproduction without cause would be biologically wasteful. Internal regulation is also necessary to signal the passage from one phase to the next at the right time. This regulation is not achieved through strict time constraints, but rather with feedback from the cell. In this podcast, we will talk about the different phases of the cell cycle. We will talk about the checkpoints where a cell is checked for certain crucial events. In that discussion, we will learn the stop and go signs. We will also talk about cyclins and cyclin dependent kinases or CDKs. The last thing we will talk about is what disruptions in the cell cycle could cause cancer. So let's get started. The first gap in the normal cell cycle is called G1. During this period, the cell must monitor both internal and external environment to prepare the DNA for replication. The duplication of DNA takes place in the second phase, which is the S phase. Note that it is called the S phase because this is where DNA is synthesized. This phase is the longest phase of the cell cycle. The second gap, also called the G2 phase, ensures the duplication of chromosomes and the division of the parent cell into two daughter cells. These processes are mitosis and cytokinesis. Also during this phase, the cell is monitored to make sure that DNA has replicated and conditions are favorable. There is a major checkpoint at the end of G2 that controls the entry into the M phase, but we will discuss that later. The last phase is the M phase. During this phase, the chromosomes are aligned and separated into sister chromatids. The M phase is separated into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. Even though cytokinesis is separated, even though cytokinesis separated the parent cell, it is usually occurring during anaphase. It is essential that the daughter cells are exact duplicates of the parent cell. Mistakes during the phases can lead to mutations that can be passed on to new cells. To prevent mutations, internal control mechanisms operate at 3 different cell cycle checkpoints. A checkpoint is one of the several points in the cell cycle where cell may be halted from going to the next stage until conditions are favorable. The G1 checkpoint determines whether it is okay for the cell to begin dividing. The cell will only pass this checkpoint if it is the appropriate size and has a reasonable amount of energy. The cell can also advance back to G0 or the inactive phase and wait for conditions to improve. If the cell meets the requirements to enter If the cell meets the requirements, it will enter the S phase and begin DNA replication. The G2 checkpoint stops entry into the M phase if certain conditions are not met. The most important role of the G2 checkpoint is to ensure that chromosomes have been replicated without mistakes or damage. If this checkpoint detects problems, the cell will attempt to complete or repair DNA. The cell is able to move to the next phase. The M checkpoint will occur near the end of metaphase. The M checkpoint is also called the spindle checkpoint because it determines whether all the sister chromatids are attached to spindle microtubules. After this checkpoint, the cell is free to split. The checkpoints that we described are established by proteins that trigger the entry and exit from phases of the cell cycle. These proteins are called cyclin-dependent protein kinases and cyclins, or CDKs. A CDK adds a negatively charged phosphate to groups to molecules by phosphorylation. CDKs are dependent on cyclins as suggested by the name. Cyclins bind to CDKs, activating the CDK phosphate to phosphorylate other molecules. Cyclins are named as such because they go through a cycle of synthesis and degradation. When cyclin binds to CDKs, they form a cyclin-CDK complex. This complex signals the cell to pass to the next phase. Eventually, the cyclin degrades and signals the exit from a phase. During cancer, the proteins involved in regulating cell division no longer signal progression from one stage to the next. Rather than lacking function, cancer cells reproduce at a rate far beyond the boundaries of the cell cycle. Cancers occur due to an alteration of the cell cycle. Cells that go through the cycle unchecked may form malignant tumors, then develop and spread through the body. Abnormal or cancerous cells grown in vitro have been transformed due to the genetic changes affecting proteins involved in the cell cycle control. These transformations have led to genes and proteins important for moving the cell forward. These genes have been named oncogenes. Scientists have combined genetic manipulation with the transformations that form genes and proteins that are important for restricting the cell cycle progression. These genes are called tumor suppressors. And that is the cell cycle and its regulations. Hope you enjoy.